So, what was so cool about this experiment is that by radioactively labeling either the protein or the DNA, it allowed Hershey and Chase to see where does this protein go and where does this DNA go. Because remember, this is before we knew that DNA was the genetic material. But after this experiment, we saw that the DNA was getting injected into the bacteria, not the protein. The DNA is actually what's going to pass on to the next generation. When the bacteria have babies, when they divide, the new generation of bacteria, the daughter cells, are going to have the DNA from the virus, not the protein from the virus. So let's think back to some kind of basic biochemistry that we learned earlier in the year. DNA and RNA are a type of biological molecule called a nucleic acid. The polymers, you could call them polynucleotides, or we could just call them nucleic acids. And the monomers or building blocks are called nucleotides. Nucleotides have three parts. They have the nitrogenous base. This is actually what gives them their individual code. The nitrogenous base has the genetic code. The nucleotide also has a 5-carbon sugar. For DNA, it's deoxyribose. For RNA, it's ribose. And they also have a phosphate group. The sugars and the phosphates together make up the backbone. So that would be the blue and the yellow part of this picture. The blue part is the sugar, the yellow part is the phosphate. The bases make up more of the horizontal part of the DNA, rope ladder shape here, and the bases are giving that genetic code. There are 4 different nucleotide bases, these little nitrogen bases: adenine, cytosine, thymine, and guanine. For RNA, there's uracil, which is U instead of thymine. RNA and DNA have a few important differences. First of all, that sugar. Second of all, that different nitrogenous base, the uracil instead of the thymine. Also, RNA is single-stranded while DNA is double-stranded. And RNA and DNA also have different functions. But this is really all stuff that we've already learned in the beginning of the year. We learned about nucleotides. We also learned about how thymine and cytosine and uracil are considered pyrimidines, while adenine and guanine are considered purines. That nucleotide structure, remember there's 3 parts: sugar and the phosphate. They make up the backbone of the nucleic acid. So you can see here some good reference pictures in the slide for you to look at as well. We also know the shape of DNA. The shape of DNA is called a double helix. It kind of looks like a twisted rope ladder. The vertical parts of the ladder would be this sugar phosphate backbone, the blue and yellow part, and the horizontal parts of the ladder with the genetic code, those are the nitrogenous bases. That shape is called a double helix. Another important thing about DNA is the base pairing. So adenine always pairs with thymine, guanine always pairs with cytosine. If an RNA molecule is pairing with DNA, then the RNA U, uracil, will pair with the DNA A, adenine. Watson and Crick are the ones who got the Nobel Prize for discovering this shape. However, there were some other scientists that contributed to this as well, such as Rosalind Franklin. She's the one who discovered this X-ray crystallography picture here that helped lead to the discovery of the shape of DNA. There's Watson and Crick.